everyone, how are you today? I hope you're okay for today. In this video, I will present about my research paper and the title Ninhydrin reaction in amino acid and protein experiment. Before starting the presentation, let me to introduce myself. My name is AMBAR and the name 44114221010. And I'm from Biology Study Program, and I'm from Universitas Negeri Semarang. Firstly, introduction. The Ninhydrin reaction is a general reaction for indicating the presence of amino acid. All amino acid reaction with ninhydrin are T-ketohydrin hydride to form aldehyde with one lower C atom and release ammonia and carbon dioxide. In addition, a blue color complex or proline and yellow hydroxyproline is formed, which is thought to be caused by two ninhydrin molecule reaction with ammonia after the amino acid are oxidized. Ammonium salts, amine, most peptide and protein react similarly without releasing carbon dioxide and ammonia. A positive reaction is indicated by the presence of a blue, yellow, and purple color. Next, I process to the object of the research is to determine the presence of amino acid in protein with uji ninhydrin. And the next, I process the research to the method. The research method used in the experimental method. The experimental method is a research method that has the aim of conducting an experiment in order to determine the cause and effect relationship that arise in a variable compared to other variables. The material used in the experimental method is ammonium solution 2%, casein 2%, and peptone 2%. And the reactor used in the experimental method is ninhydrin solution 0.1% and ammonium sulfate. And the tool used in the experimental method is a drop pipette, beaker glass, test tube, and graduate cylinder. And the next are experiment method. The first step that was taken was to put 2 milli of material and 0.5 million ninhydrin react into the reaction test tube. And then put in a water better from 10 minutes. And finally, watch the color change in the solution. It is blue, yellow, or purple. My experiment results are present in table form. Before discussing the result, you need to know about the positive result of amino acid with the ninhydrin test. Marked by change in the color of the solution become blue, yellow, purple, or red brown. The first test tube containing 2% albumin and ninhydrin reagent products a blue solution, which means the tube is positive for amino acid. And the second test tube containing ammonium sulfate and ninhydrin reagent product is a transparent, which means the tube is a negative for amino acid. And third, containing casein 2% and ninhydrin reagent product is yellow, which means the tube is positive for amino acid. The last, peptone 2% and ninhydrin reagent products a purple solution, which means the tube is positive for amino acid. And the next, the discussion. The was explained in the introduction section that the working principle of Ninhydrin test is to be the presence of upsurge of protein in contained by ending Ninhydrin reagent to determine the amount of free amino acids contained there, where free amino acid will be contained by Ninhydrin reagent and form a color complex. From the experiment that have been carried out, different color complex are formed in each sample piece. The color difference product in the experiment carried or were caused by difference in the amino acid group. Positive result in the experiment were indicated by a change in the color of the sample to blue, which means that the sample contained the amino acid group proline, and to yellow, which means that the sample contained the amino acid group hydroxyproline, and the last to purple or reddish brown, which means that the sample contained the amino acid group decatonhydrin. Sample that do not change color to blue, yellow, or purple can be said to be a negative for amino acid. The negative result is because the sample used cannot reaction with Ninhydrin reagent. For example, in the experiment, the ammonium sulfate sample product a transparent color. This is because ammonium sulfate is included in the ammonium salt group. When the ammonium salt does not contain amino acids, so it cannot react with the Ninhydrin reagent. From the result and discussion, it can be conclusion that first, albumin 2% show positive result containing amino acid, which were marked by change is colored to blue, which means that the sample contained the amino acid group proline. Second, casein 2% show positive result containing amino acid, characterized by a yellow color change. Which means that the sample contained the amino acid group hydroxyproline. And third, peptone 2% show positive result containing amino acid, which is characterized by a change is colored to purple, which means that the sample contained the amino acid group decatonhydrin. And the last, ammonium sulfate show a negative result that does not contain amino acid, which a marked by change is color to become transparent because ammonium sulfate include the ammonium salt group. When the ammonium salt does not contain amino acids, so it cannot communicate with the Ninhydrin reagent. That's the presentation of my research paper. Thank you.